Science Matters. The topic for today is the gut microbiome, mild cognitive impairment, and probiotics. Fascinating recent double-blind placebo-controlled study is just another example, we think, of how the microbiome plays a critical role in overall body homeostasis and, as you'll see in this study, acts as an important component in brain health. And I will, with that, let Dr. Aron Gonshor take it away.
Well, thank you, Reed. Good to see you again. It's good to be
Nice
back.
to see you.
And as we say each time in each one of these sessions, we take a deep dive into recent research to bring to light objective scientific evidence of whatever the topic at hand happens to be. And today, we're going to look more closely at the gut microbiome, mild cognitive impairment, and probiotics. And we'll discuss what each one of those things means. But this study, like other recent research, is beginning to show objective evidence of the significant role that's being played by both pro and prebiotics in cognitive function. So let's look at the actual evidence, because science matters.
Well, fantastic. And I'm really looking forward to this. It's a good study. But I have to say, Dr. Gonshor, on our previous broadcast of base editing and decreases in LDL cholesterol and cardiovascular disease, that stimulated quite a few questions. And a very common thread was a question of where the science of RNA stands today, both as a research tool and its use in clinical practice.
Well, this is an incredibly important series of questions. This is going to be a longer answer because we're bundling a number of questions together. There are essentially two technologies that I think retrospectively are going to be seen as critical players in what one can consider a fundamental turning point in medical treatment. One of them is artificial intelligence. And, there's been a lot of attention today, lately, on artificial intelligence, and rightly so. It will definitely change things. So stay tuned. We're going to be, I'm sure, discussing AI and some of the effects it has. But the other technology is a bit more obscure, but I suspect it may be just as impactful or more so. And what I'm talking about is RNA therapeutics, which many are now considering one of the cornerstones of future medicine. Many diseases are fundamentally a problem of proteins. In some cases, like what we discussed the last time around, which is hypercholesterolemia, which meaning a lot of cholesterol in the blood, the body produces too much of this protein cholesterol. So we highlighted the use of CRISPR technology and messenger RNA technology and nanoparticles, etc., as part of what's called base editing, that technique. And in fact, as a follow-up to what we talked about last time, on November the 17th, there was an announcement by that company Vertex, the one that gave that presentation at the American Heart Association, that it had received approval in the United Kingdom to market the first CRISPR drug, not for cholesterol, but for the treatment of sickle cell disease. And for those who know anything about that, it's a giant breakthrough that is going to be helping millions. And just this past week, I literally just put this into my notes. There was a study published on base editing, and it was in the Nature Biomedical Engineering journal, and it was called, it's a long title, Optimization of Base Editors of the Functional Correction of SMN2 as a Treatment for Spinal Muscular Atrophy. Now, what does that mean? Well, Spinal muscular atrophy, SMA, it's a devastating neuromuscular disease that remains a leading cause of infantile death worldwide. Now, SMA is primarily characterized by what? The death of motor neurons in the spine. Because of that, there's muscle deinnervation. Muscles do not get innervated by the nerves. And because of that, you get muscle weakness, incredible muscle weakness. Most SMA cases are caused by a loss of function mutation within these motor neurons, their genes. So in this very detailed paper, the authors concluded that targeting these mutations via base editing or other CRISPR-based methods, that's their words, may provide long-lasting outcomes to patients with SMA. I come from a neurophysiology background. I'm a PhD in neurophysiology. And I can tell you, we didn't even dream of anything like this when I was studying this.
Sure.
Now it's coming true. So the speed with which this new technology is advancing therapeutics, and may I say possibly, quote, cures, is nothing short of breathtaking. All this would be exciting and exciting enough, but as you might hear in the, these commercials you get for kitchen utensils on TV, they said, wait a minute, there's more. And in this case, we're talking about a second type of RNA usage. Enter into the picture small inhibitory RNAs, siRNAs. They're the body's way of targeting proteins for destruction before the protein ever gets created. These little RNAs are about 20 base pairs long, very, very short. And they seek out a complementary target messenger RNA attached to it. They glomp onto it, and they call in a group of proteins to destroy this protein before it can actually get started. So with the target mRNA gone, no protein can be created. In fact, on November 12th, the same day that the CRISPR mRNA work was highlighted at the American Heart Association, an article was published in JAMA, and it was titled Lepodisiran, an extended duration, short interfering RNA targeting lipoprotein A. Long title. What is it? A randomized dose ascending clinical trial. The lipoprotein A, by the way, is a LDL-like protein. That's the bad cholesterol. And it's associated with cardiovascular disease, with heart attack, with stroke. After a single injection of this particular drug, the lipoprotein A levels started to drop within a week of administration. And then, get this, they stayed down. In the higher dose group, levels were nearly undetectable a year after the injection. So a single injection that can suppress protein synthesis for an entire year, if it really works, and again, we're just in the beginning of this. We're making baby steps. If it really works, this is a game changer, and I'm not using that term lightly. But wait, there's more. This past July, the New England Journal publishes a study on zilebesiran. I hope I'm saying it right. It's another one of these small inhibitory RNAs that inhibits the production of angiotensinogen. I remember learning about that when I was a student. It controls blood pressure. Similar story. One injection led to basically a complete suppression of angiotensinogen and then a sustained decrease in blood pressure. So let's dream a little, because that's what these authors were doing. There may come a time in the not too distant future when we go to the doctor once a year, we get an RNA shot, don't have to take any other medication from that point on. And some researchers are noting, get this, that may happen as soon as 5 years from now. Now, if that's true, that is powerful.
It's quite a consideration for sure. And the thought of shots scares me, but the idea of for people who are sick, not having to take as much medication is promising. Also, when you mentioned the sickle cell anemia, that disease, if it could be tweak a gene and you don't have it anymore, that's better than the alternative for sure. It's and millions of people could be helped, like you say. So I have a careful and watchful eye in this kind of a thing. And I think it's fantastic that we're talking about it, Dr. Gonshor, and that was a good explanation. And I want to thank everyone for submitting those questions. And we're all learning together. Dr. Gonshor, I want you to jump into the study, but I want to say to everyone that you're submitting your questions, include any comments and subjects you'd like to see, directions. This rather new, it's still a newish podcast and show, so we do need feedback, so please feed something into us and then we can respond to that. So Dr. Gonshor, let's jump into today's study about the gut microbiome, mild cognitive impairment, and probiotics.
Hello, hello, hello. Well, today's study, the gut microbiome, which we've talked about in previous sessions, in this case, we're going to be relating that to mild cognitive impairment and probiotics. And the study came up recently, I think just the end of last year. And what they were saying is that there are specific members of the gut microbiota. Remember, that's the 15 trillion or who knows how many trillion bacteria in the gut are correlated with cognitive performance in middle-aged and older adults. If this can be replicated, it could be a key early indicator of mild cognitive impairment and manipulated by probiotics, prebiotics, and symbiotics to promote successful cognitive function while aging. So what does all this mean? Well, let's give you a little bit of background. Remember that the gut has its own brain. There are about 500 million nerve cells, 20 neuronal types, various autonomous microcircuits, which allow the gut to be doing its own functioning without having to speak to the brain continually. It has chemical and mechanical sensing, and of course, it controls muscle movement in the gut, etc. And the gut-brain axis, which you hear an awful lot about, is based on the fact that the central nervous system will integrate a lot of information, behaviors, etc., can send signals down to the gut. The gut, on the other hand, by doing the digesting, by having the immune defense function, and so on, can send signals to the brain. I'm mentioning two here in particular, which are very well known. Leptin, which is a signal for satiation, meaning you say, I've eaten enough and that's it. I don't want to eat anymore. You tell the brain to not worry about wanting to have you continue eating. And ghrelin is just the opposite. It lets the brain know that you're hungry and you need to go out to forage. You need to go out and find food. So that's just a simplistic way of talking about that. We do know with the gut-brain axis that in the gut, there are sort of three large areas that it involves itself when it comes to the brain. One is peripheral serotonin. As you probably know, gut cells produce large quantities of tryptophan, which then forms serotonin in the gut, and that affects brain signaling. There is the immune system, in which the microbiome prompts immune cells to produce cytokines. Those are signalers, and those signal the brain to do whatever it happens to want to do at that particular moment. And the third large category are bacterial molecules. Microbes produce metabolites. In this particular case, we're talking about a short-chain fatty acid like butyrate, which a lot of bacteria produce. And butyrate, amongst many things that it does, is able to fortify the blood-brain barrier by tightening the cell connections, etc. So it's just to give you a quick primer on that, to tell you that there really is an issue of the gut-brain axis. The rest of the background context is to remember that inflammation has a major effect. Dysbiosis, or an imbalance in the way the bacteria act within the gut, can cause chronic inflammation. And this can be a key part of things like Alzheimer's disease. So inflammatory molecules are going to disrupt neural function, and they can contribute to the formation of amyloid beta plaques. And amyloid plaques are really considered one of the hallmarks of Alzheimer's disease. It can be involved in metabolic changes. The gut microbiome affects metabolism, specifically lipids and glucose, and metabolic dysfunction is known as a risk factor for things like Alzheimer's disease. So the microbiome changes contribute significantly to these metabolic shifts. And last but not least of these three things is the alteration of intestinal permeability. And dysbiosis, we know, can lead to an increase in this permeability, which allows harmful substances to enter into the bloodstream and potentially into the brain, which contributes to the progression of Alzheimer's disease. In this present study, we're not looking directly at Alzheimer's, we're looking at people that are being evaluated for their cognitive status. So they took 169, 170 people. They were aged between 52 and 75 years of age. This is a double-blind, placebo-controlled, randomized clinical trial. This is as good as it gets. And for 3 months, the participants in this study received either a probiotic containing a specific strain of bacteria, in this case, Lactobacillus rhamnosus, very well known, or they received a placebo. And all the participants underwent an in-depth analysis of their intestinal microbial constituents at the beginning and the conclusion of the study. And when you actually read the study and you see how they did this in depth, is speaking mildly. They really go through it in detail. And what were the findings? They found with microbiome analysis that they were able to identify some specific taxa that were correlated to mild cognitive impairment, MCI. What were they? Well, there were 3 in particular: Prevotella ruminicola, and 2 types of Bacteroides, Bacteroides theta and Bacteroides excellenosolvens. The Bacteroides theta is very, very well known. I'm not going to say the entire name. I can never say it properly. And in those participants that had those specific taxa, it was characterized as they were having mild cognitive impairment at the beginning of the study. They received a probiotic, and there was an improvement in their cognitive function. So the mild cognitive impairment, remember, is an early stage of full-blown Alzheimer's disease and is a marker of progressive cognitive decline. So Prevotella ruminicola and the Bacteroides theta in particular were identified as taxa that could be modulated with pre, pro, and symbiotics. And remember, symbiotics is a mixture of both the probiotic and the prebiotics. And so with the pre, pro, and the symbiotics, was able to prevent or slow the progression of cognitive impairment. So what are the key takeaways here? There are specific members of the gut microbiome that correlate with cognitive performance in middle-aged and older adults. Specifically, Prevotella ruminicola and Bacteroides theta, these are taxa that could be modulated with the pre, pro, and symbiotics to prevent or even slow the progression of cognitive impairment. Should these findings be replicated, and of course you need to do many more studies, these taxa could be used as key early indicators of mild cognitive impairment, and they can be manipulated by the use of probiotics, prebiotics, and symbiotics to promote successful cognitive function while we age. So this was a short review of the study because we spent a lot more time on going over some other things. But I thought that it was a very interesting study to, again, show you the effect that the microbiome has in functioning of all parts of our metabolic universe in our body.
Well, fantastic. Great study. And it's not a big surprise that the microbiome is so important in all of the other metabolic processes, and we certainly want to improve people's gut health. It's kind of what we do. But seeing these studies that back up everything we say and do is really, really critical. And especially around this taking probiotics, because I think every FDN uses them a lot. And we also do a lot of testing. I find it interesting that there are these three different bacteria, Dr. Gonshor. Since you said trillions of bacteria, how do they sort that out? Are these tests that anyone could do, or where does that fit in?
I think one of the things we need to recognize is that we're making the first baby steps. There's so little that we understand, not just about individual bacterial colonies, taxa, and so on. What we also don't understand is what happens when they are acting in consort, when they're acting together. When you put them together, then it's not symbiotic, it is what's the term that we use? It basically creates a greater than the whole, because altogether they do even more than they would do individually. And so we don't even begin to understand yet what are the various types of groupings of bacteria that need to come together, not just individual ones or even two or three. It may be that you need dozens before the real effect begins to show itself. That's why I would humbly say that, notwithstanding that there's some beautiful work being done now in these studies, and also there are companies that are beginning to offer a look at various types of bacterial colonies to tell you what's going on. I would humbly submit we're not there yet. We're getting there quickly, but we're not there yet. Does it mean that everything they're showing means zilch? No. But there's a lot more to come, and it's going to come very, very soon, where we're going to be able to say, here is a grouping of 10, 20, 30 different taxa that when you put them together, now the magic begins to happen. We're not there yet.
Yeah, I really love this discussion, and we need to sort of move on and let people go. The Science Matters was designed to be a 15-minute program.
Yeah.
But and thanks for sharing the answer to the question about last week's presentation. It was clarifying, and I think you deserve as much time as needed to go over that. And today's presentation, no doubt, will have stirred up some questions. Again, lots I'd like to talk about it. But I'll just make this one comment. The tie-in, there's FDNs who are very natural. So a study like this is perfect because it tells you there are specific probiota that will help in these certain conditions. As a matter of fact, all of life, in a sense, depends on it. But so we want healthy microbiota, and then we can sort of drive the industry, because if we tell the labs, look, this really important research says these are the bacteria we want to identify, then labs they'll start figuring out a way. They want to serve, and so it's a nice cycle. We got the research backing up what we do and helping influence improvements in the entire industry, I think. So good work on everybody. And obviously, if you like this episode, please subscribe, like it, share it, and submit your questions for this or any other episode, any comments and suggested topics. That's very important. We'd like to know where we should take this. Submit those to fdntraining.com/science matters. fdntraining.com/science matters. And if you want copies of the studies that we use, you can get them directly from fdntraining.com/studies. They're all in there. We're on episode 15, Dr. Gonshor. I think that's fantastic. And want you all to recognize that and see us again in 2 weeks. January 10th, it'll be the new year, right, Dr. Gonshor? Anything to sign off the year with? I think we had a good run at it, and we'll continue next year. The other thing I'd like to say is that if you guys are hanging out with our tribe, fantastic. There's a Dr. Gonshor and I are getting together on the first Saturday of January, which I think is the 6th. And we're going to talk about these studies and, you know, which ones you could trust, how you can trust them, how you could get them yourself and analyze them. So that'll be a pretty important thing. Independent of this, Science Matters is our health space unmasked. And Dr. Gonshor is our guest. Please look that up, and we'll see you next time.
And just a reminder, Reed, that on January 10th, we're going to be discussing metabolic efficacy of time-restricted eating. A lot of people have asked us about this. Time-restricted eating in adults.
Wonderful.
And we're going to do that because
science
matters.
matters.